Okay, let's have a look at question 1. Here they give us figure 1.1, which is a diagram of an animal cell. Question A then asks to complete table 1.1 by writing the name and function of each of the labeled parts of the cell shown in figure 1.1. So let's look at the different cell organelles that we can find. So, first in animal cells, we know that we get a cell membrane as well as cytoplasm. Now, the cell membrane has the function for selectively importing or exporting any type of chemicals. So, this cell membrane will be semi-permeable and certain substances will be able to enter the cell or exit the cell. Now, the cytoplasm of the cell is where all the chemical reactions or metabolic reactions take place within the cell. Now, a plant cell has an additional cell part or cell organelle called the cell wall. Now, the function of the cell wall is to give structure and stability to the cell. Now, inside the cell, we can also find organelles like the mitochondria. Now, the mitochondria is responsible for aerobic respiration, which creates ATP, which is a currency for energy. Now, the mitochondria can be identified by these indentations called crista, as it has a double membrane. We can then also find chloroplasts. Now, these organelles are mainly in plant cells. They're responsible for photosynthesis and the production of carbohydrates like glucose. Now, chloroplasts can be identified by these flattened stacks that are packed upon each other, giving this dark appearance called grana. We then also have a nucleus, and the nucleus is found in all eukaryotic organisms. Now, the nucleus has a nuclear membrane, also has a nucleolus, and then nucleoplasm. The function of the nucleus is for the storing of our genetic material. It's also responsible for the manufacturing of ribosomes. We then have the Golgi apparatus, and we won't have too much to do with them in IGCSE. So the Golgi apparatus has a function of sorting and then exporting chemicals and substances out of the cell. We then have the rough endoplasmic reticulum, and this is identified by tiny little dots called ribosomes on top of it. The rough endoplasmic reticulum is responsible for the manufacturing of proteins. Lastly, we have the vacuole. Now, the vacuole is mainly found in plant cells. Animal cells also have a vacuole, but it's much smaller and it's called a lysosome. Now, the vacuole is identified as a large empty space, but in reality, it's not an empty space. It's filled with water as well as nutrients and other biological substances. Okay, so let's answer the question. So A, which is the big black dark dot, would be the nucleus. Now we know that the function of the nucleus is to store genetic information, or otherwise known as DNA. We then have B. Now we can see that B kind of is indicative at this empty space inside of the cell, and this is called the cytoplasm. So this is where metabolic reactions take place. C then indicates the outer membrane of the cell. So this is the cell membrane, and this controls what goes in and out of the cell. Lastly, we have D, which is this tiny organelles, and we can see that they have indentations. This is the mitochondria. So remember, the mitochondria provides energy through aerobic respiration. Next, we have question B. First, they're asked to explain how water moves into and out of animal cells. Now, we know that water moves through a process called osmosis. Now, osmosis is the process where water molecules move through a semi-permeable membrane from a region of higher water potential to a region of lower water potential. So let's say we have a beaker, and within this beaker is a solution with a water potential of negative 0.77. In the solution is also red blood cells, and they have a water potential of negative 0.77. Because the water potential in the red blood cells are equal to the water potential in the solution, water would move out of the cell at the same rate as water moving into the cell, meaning that there's no net movement of any water molecules. Thus, the cells will neither shrink and they will also not become swollen. Now let's say we have a second beaker, and in our second beaker we have a very salty solution with a water potential of negative 25. We then take the red blood cells and we add it to the solution. Now remember, the red blood cells has a negative 0.77 water potential. This means the water will move from a higher water potential to a lower water potential. So it will move from negative 0.77 to negative 25. This means that water will be lost from the red blood cells and they will shrivel up. In our third beaker, we have pure distilled water. This means it has a water potential of 0. If we add our red blood cells to the solution, the water will move from the higher water potential, which is the solution, to the lower water potential, which is the red blood cells. So water will move into the red blood cells, and the red blood cells will swell and eventually burst because they cannot handle the pressure. So let's get back to our question. The first part of question B asked to explain how water moves into and out of animal cells. Now, water moves into and out of the cell by a process called osmosis, whereby water moves from a high water potential to a low water potential. So this will be down a water potential gradient, and it will also move through a semi-permeable membrane. Now, the second part of question B says that a sample of red blood cells was taken from a person. The red blood cells were put into 3 test tubes. Each test tube contained a different liquid. So this is similar to what I explained in my example. So, the first one was blood plasma. This would be where the liquid has the same water potential as the blood, and then also pure water, and then a very concentrated salt solution. Now, figure 1.2 shows the appearance of red blood cells when examined using a microscope. Identify the liquid the red blood cells were immersed in. Write your answer in the spaces provided in figure 1.2. So if we look at the first figure, we can see that this illustrates normal blood cells. We can see the biconcave shape. It kind of looks like a folded donut. It's normal, meaning that this blood would have been found in the blood plasma test tube. The second image kind of shows this shriveled up spiky blood cells. These are not normal blood cells, they're abnormal. They're shriveled up, meaning that they have lost water. This means that they would have been found in the salt solution because water would have moved out of the cell into the very concentrated salt solution. Now, the third image that we have is an image of burst blood cells. Now, if you followed my example, you would know that they burst because too much water moved into the cell and they cannot handle the pressure. This means that the cell was probably found in the pure water sample. Now, a common mistake that can be made with this question is when students look at the second figure and they see that the blood cells are very round. This might make them think that the cells are swollen and that they were found in pure water, but this is not the case. You can avoid this mistake by looking at the other 2 images. We know that we have 3 solutions. We have blood plasma, pure water, and a concentrated salt solution. Now, the first one is normal cells. It's easily identifiable. And the last one is also very identifiable because it's a normal blood cell, but it's burst open. So using a method of deduction and elimination, we can already say that the first image was blood plasma, the last image was pure water, and then the only one left would have been the very concentrated salt solution for the middle picture. So on the next page, we have a couple of questions on different blood cells. So before we start answering these questions, let's go through the different blood cells and their functions. The first blood cell that we'll have a look at is our red blood cells. Now, our red blood cells have this biconcave shape. It kind of looks like a donut, but the center isn't punched out all the way. The reason why it has this shape is because it doesn't have any organelles or a nucleus present in the cell. This is to free up space for hemoglobin. Now, hemoglobin is a protein molecule which is responsible for the transport of oxygen. Oxygen is able to bind to this molecule and be transported to all your cells. We then have lymphocytes. Lymphocytes is one of our white blood cells. They have a very large round nucleus. They are responsible for the production of antibodies. Antibodies are able to attach to specific cells, more commonly foreign cells, and tag them to be destroyed. Now, the last cell that we're going to look at is a phagocyte. Now, a phagocyte is also a type of white blood cell. Now, the phagocyte can be identified by its nucleus that has an irregular shape or is lobed. They also have a more granular cytoplasm. Now, when the antibody tags a cell to be destroyed, the phagocyte will actually go and destroy the cell by engulfing it and using different enzymes to break it down. This process is known as phagocytosis. Okay, let's have a look at our questions. So the first question on this page says to state the name of the molecule in red blood cells that combines with oxygen. Now we know that this molecule is called hemoglobin. We then have figure 1.3 that shows a drawing of a white blood cell. So we know that it's either a monocyte or a phagocyte. They then ask to identify the type of white blood cell that's shown in figure 1.3. Now, if we look at this white blood cell, we can see that the nucleus inside of the cell is lobed. This means that it would be a phagocytic cell. Remember, the nucleus of a monocyte is perfectly round. On the next page, we have question 2. It states that figure 2.1 is a photograph of a spider plant, Chlorophytum cornutum. Now, spider plants can reproduce by producing an identical plantlet, which grows away from the single parent plant. We then see the image of the spider plant and the little offspring plantlet. Question A then asks to identify one feature in figure 2.1 that shows that spider plants are monocotyledons. So let's quickly go through the differences between monocotyledons and dicotyledons. So the first difference between a monocot and a dicot is in the seed itself. So a monocotyledon plant has only one cotyledon in its seed, whereas a dicotyledous plant has two cotyledons in its seed. Now, if you don't know what a cotyledon is, it is part of the embryo within the seed of the plant. So this means that it can become the leaves of the seedling or the roots of the seedling or anything in that regard. The next difference that we are going to look at is the stem of the plant. Now, if we look at the vascular bundles in the monocot stem, we can see that they're randomly arranged and there's no pattern followed for the arrangement. But in a dicotyledous plant, the vascular bundles are arranged in a circle, kind of on the outside of the stem. Then we also have the leaves. In a monocot plant, the veins on the leaves are parallel, like you can see in this image, whereas on a dicot plant, they have branched leaves. Then we also have the roots, where monocots have fibrous root systems and dicot plants have a taproot system with tiny roots coming off of the taproot. Lastly, we have our flowers. Now, a monocot plant has flowers that have petals in the multiples of 3, whereas a dicot plant has flowers that have multiple petals of 4 or 5. So now we can answer question A that asks us to identify one feature that shows us that it's a monocotyledon plant. So one feature that can be seen easily is the fact that the plant has narrow leaves with parallel veining. The second question then asks us to describe the advantages and disadvantages of asexual reproduction in a population of spider plants in the wild. So let's look at what is asexual reproduction, and then we can look at the advantages as well as the disadvantages. The first example of asexual reproduction is binary fission. Now, binary fission most commonly occurs in bacteria, where the bacterial cell will grow, it will multiply its DNA and its organelles, and then it will split into 2 different bacteria that are genetically identical to one another. The second type of asexual production that we get is vegetative propagation, and this is usually done by humans. In vegetative propagation, a piece of the plant, like the succulent especially, can be removed from the parent plant. It is then planted into the soil, and a new little plant will grow from it. These 2 plants will also be genetically identical to one another. Then we have runners, like in the spider plant that we saw, but we can also see it in strawberries. So what happens with a runner is a little stem will grow, and when it finds soil again, it will create a tiny plantlet. Now these plantlets, just like all the other asexual reproduction examples, are also genetically identical. Then we have budding. Now, budding can be seen in potato plants. Now, in potato plants, they create these tubers of potatoes, and these tubers will grow new roots and new shoots. And when you plant them, you'll have a genetically identical potato plant that comes up. Okay, so let's have a look at the benefits and the disadvantages of asexual reproduction. Now, firstly, we know that the offspring are genetically identical. So if the parent is well suited for the environment, then the offspring will also be well suited for the same environment, making this property a benefit to the plant. But it will also mean that diseases are more likely to affect the whole population, as all of the offspring will be susceptible to the disease. The second is there is no fusion of gametes that's needed, because all of the offspring will be genetically identical to the parent plant. This means that no male is needed, or there wouldn't be need for a second plant. This also means that less energy is used, and asexual reproduction is more energy efficient. It is also much faster, as the reproductive mate isn't needed, so the plant doesn't need to search or wait for a reproductive mate. Now, the disadvantages of this is that evolution will occur much slower, because gametes don't fuse and genetics don't mix. Then lastly, we have that the offspring will grow close to the parent plant. This means that the population can exploit the environmental conditions. So if it's a favorable environmental conditions, the plants will grow extremely fast. It also means that there will be more competition for resources between the plants, and some plants might die off because there's not enough nutrients or water or even sunlight in their environment. Okay, let's read the question again. They ask us to describe the advantages and disadvantages of asexual reproduction of population of spider plants in the wild. Advantages would include that it is faster than sexual reproduction. It also does not require a male or fertilization to take place. Thus, it will be more energy efficient. The offspring, which would be genetically identical, will be well adapted for the environment. Now, the disadvantages include that there will be less genetic variation, and thus the population will be more susceptible to diseases. There will also be more competition between the offspring and the parent plant, as the offspring will grow close to the parent plant. On the next page, we have question B. It states that figure 2.2 shows pollen that has been released from the flowers of a hazel tree called Corylus avellana. These flowers are wind pollinated. Before we start with the questions that follow, let's look at different pollination methods. The two pollination methods that we'll be looking at is wind pollination, and here we will use maize as an example, and then also insect pollination, and here we're just going to use a normal flower as an example. If we look at the maize, we can see that it has two types of flowers. The first is a male flower and the second a female flower, whereas the flower is both male and female parts together. Now the flower is brightly colored to be able to attract insects, whereas the maize has no bright colors because wind doesn't care what color the plant is. In the maize, the anthers, the male part that has the pollen on it, hangs outside of the plant. This is so that wind can sweep the pollen up into the air. In the flower, the anthers are small and inside the plant. This means that the insect needs to be able to crawl into the plant to get pollen on its stomach and its legs. On the wind-pollinated flower, the stigma hangs outside of the plant and is kind of feathery. This is so that pollen that is swept up by the wind will be able to get trapped in this feathery stigma outside of the plant. For the flower, the stigma is small and inside of the flower as well. This is for when the bees crawl inside the flower so that when they want to get nectar, they will rub the pollen off of the stigma in the flower. Now, wind-pollinated flowers produce a lot of small pollen. This is to ensure that enough pollen reaches the same species of plant through the wind. Remember, wind doesn't know what plant is the same species, which means that it all happens by chance. Now, the flower produces less pollen, but these pollen grains are larger and they're spiked or even sticky. This helps the pollen to stick to the legs and the abdomen of the bee, which means that when the bee flies away, the pollen won't just fall off. Another difference between these two pollinated flowers is that the insect-pollinated flower may produce a scent or even nectar. This scent will be able to attract bees, and the nectar will feed the bees. The bees can use the nectar to make honey, whereas the wind-pollinated flower does not need any of this, as wind does not care. Wind is not a living thing, so it cannot smell or eat at all. Now, flowers can either undergo self-pollination, where pollen from the plant falls on the stigma of the same plant, or they can undergo cross-pollination, where the pollen from one flower is transferred to the stigma of another plant in the same species. Once the pollen falls on the stigma, then the same happens in both cross-pollination and self-pollination. So what happens is when the pollen grain lands on the stigma, this pollen grain will release enzymes. The enzymes will then cause a pollen tube to grow down the style. The nuclei of the pollen grain will move down this tube all the way to the ovule. So this nuclei, being the male gamete, will then fuse with the nuclei of the female gamete, and this will form a zygote inside of the plant. This zygote will then form a tiny little plantlet that will produce seeds and a fruit surrounding the seeds. So now we can answer the questions that follow. So the first question asks to explain why a hazel tree has to produce very large amounts of pollen. So we know that by producing very large amounts of pollen, it increases the chance of the pollen reaching a stigma. The second question asks to describe how a stigma from a wind-pollinated plant is adapted for pollination. So the wind-pollinated stigma would be large and feathery. It will also hang outside of the plant or outside of the flower to be able to capture that pollen carried by the flower. On the next page, we have question C that says that the hazel tree uses cross-pollination rather than self-pollination. Describe cross-pollination. So we know that cross-pollination involves the transfer of pollen grains from the anther to the stigma of a different plant of the same species. In this question, it's very important to end it with the same species. If the pollen grain happens to fall on the anther of a different species, nothing would happen as they won't be genetically compatible. Question D then asks to outline the events that occur in the flower after pollination. So once pollen grain lands on the stigma of a flower, it will release enzymes that will then cause a pollen tube to grow down the style. The nucleus of the pollen grain will move down the pollen tube into the ovule. The male nucleus will then fuse with the female nucleus to form a zygote. So this happens in both cross-pollinated as well as self-pollinated plants. We then have question 3 that says that proteins are biological molecules. Explain how proteins are made. So let's have a look at how proteins are made. This by which proteins are made is called protein synthesis, and it happens in each and every living cell, including eukaryotes and prokaryotes. We're going to use a eukaryote as an example. So in a eukaryote, we have the nucleus. Now the nucleus is made up of a nuclear membrane, and all the DNA is found inside of this nucleus. The DNA in the nucleus is in the form of chromatin, which is tightly coiled DNA around histones. Now, our DNA is our body's way of coding the information for our body to use to make proteins. So we use a guanine, cytosine, thymine, and adenine code. So let's say there's a gene and this gene has CGTAAT. So it's double helix, so the mirror image of this code will also exist. Now this DNA will then be transcribed into mRNA. So the GCATTA code will turn into a CGUAAU code. Now remember, guanine always attaches to cytosine, cytosine to guanine, thymine to adenine, and adenine will attach to uracil in RNA because RNA is uracil instead of thymine. The mRNA molecule will then exit the nucleus through one of the nuclear pores and go towards a ribosome. At the ribosome, this is where protein synthesis or translation takes place. So my GCUAAU code will then attach tRNA molecules with a complementary code. So in the mRNA molecule, the three bases together are called a codon. The tRNA will have an anticodon that is a mirror image of the codon. Each anticodon is attached to a specific amino acid. The tRNA molecules will then line up on the mRNA molecule, and the amino acids will attach to each other, forming an amino acid chain. This amino acid chain will then be modified and shaped into the protein molecule. This process here is called translation. Okay, so let's have a look at how we can answer this question. So the gene that codes for a specific protein is found on DNA in the nucleus. This gene is transcribed or copied into messenger RNA called mRNA. Now mRNA then moves out of the nucleus towards a ribosome. As the mRNA moves through the ribosome, tRNA attaches to it and assembles an amino acid chain. This amino acid chain can then be modified and folded into a functional protein. The specific sequence of amino acids will determine the function of the protein. On the next page, we have question B. It says that figure 3.1 is a diagram of enzyme 1 catalyzing the breakdown of molecules A into two smaller molecules B and C. So here we can see molecule A is a triangle. It fits into enzyme 1, the circle, and it breaks it down into molecule B and C, which is then the broken triangle on the right. They then say molecule D, shown in figure 3.2, is added to the mixture shown in figure 3.1. This causes the rate of production of molecule B and C to decrease, meaning that the enzyme won't be able to break down molecule A as quickly as it did in the first reaction. On the next page, the first question asks, using the information in figure 3.1 and figure 3.2, suggests why the rate of production of molecule B and C decreases after molecule D is added. So we can go back to the previous page. So here we can see that the enzyme where the molecule fits into has a specific shape. This is called the active site, and the shape of the active site is very important because the molecule A, which is the substrate, fits perfectly into the active site. In biology, we call this the lock and key model. Now we can see molecule D on figure 3.2 has a similar shape to the substrate, meaning that it can also fit into the active site. What this means is that molecule D is an inhibitor. Molecule D will attach to the active site of molecule A. This will cause the active site to be occupied, and molecule A will no longer be able to attach and be broken down into molecule B and C. So to answer the question to suggest why the rate of production of molecule B and C decreases after molecule D is added, we can say that molecule D has a similar shape to the molecule A, thus being complementary to the enzyme active site. Molecule D will be able to fit into the active site of the enzyme, obstructing molecule A from binding. If molecule A cannot bind, then molecule B and C will not be produced, as molecule A will not be broken down. This question says that figure 3.3 shows enzyme 1 after a change in its environmental conditions. So we can see that the active site went from a triangular shape to more a staggered X shape. They then asked to suggest two changes that could cause the effect on enzyme 1 shown in figure 3.3. Let's have a look at our active site shape. So we know originally the enzyme had a triangular active site that would kind of look like this. Now each of the dots here represent an amino acid, and these amino acids are connected in a chain to give the shape. Now between the amino acids there's also different types of bonds that give the specific shape to the active site. Now the first condition that could alter the active site is temperature. Then we also have pH that can alter the site. At low temperature, the shape of the active site won't change, but the rate of reaction will change as kinetic energy decreases. But at high temperatures, these bonds that hold the amino acids together in the specific shape breaks down, and the shape of the active site would be lost. This is called denaturing. Now with pH, when the pH is either too high or too low, it bends and alters these bonds, keeping the amino acids together. When the pH increases too high or too low, it can even cause the bonds to break and it would be irreversibly changed. So the two changes that could cause the effect on enzyme 1 shown in figure 3.3 will be either a too high of a temperature or too high or too low of a pH. On the next page, we have question 4 that says characteristics of plants are controlled by genes. Alleles are alternative forms of a gene. Now remember, alleles are like the gene for eye color. One allele codes for blue eye color, and another allele will code for brown eye color. Question A then asks to explain how new alleles are formed. Now, new alleles are formed through mutation, which is a change in the base sequence of the DNA. There are base sequences, which is guanine, cytosine, thymine, and adenine. When one of those or more than one is changed, the gene changes as well. Now, mutation can be caused by ionizing radiation or other mutagens, such as chemicals. Question B then says that the number of hours of daylight affects when plants produce flowers. This ensures that flowers are produced at an appropriate time of year for each species. The response to the number of hours of daylight is controlled by genes. Wheat is a crop plant. A farmer wants to produce a type of wheat that flowers when there are fewer hours of daylight. Describe how the farmer could selectively breed this type of wheat. So in this question, it's very important to note that they ask for selective breeding and not genetic modification. So let's have a look at how this farmer could do this. So the first thing the farmer needs to do is to plant several different seeds of wheat. He will then watch them grow and give them a certain amount of daylight hours. We'll start very low at 5 hours of daylight. When no plants flower with this little amount of daylight, the farmer can increase the daylight slightly, let's say 6 hours of daylight. If still no flowers flower, then we can start with 7 hours of daylight. As soon as some of the plants start flowering, the farmer would get rid of the plants that did not flower and only keep the plants that flowered at the little amount of daylight. He would use these plants and then cross-pollinate them to produce new seeds, and he'll restart the experiment. He'll plant the seeds and give them a little amount of daylight, and then those who flower early will then be chosen, and the experiment will be repeated over and over again. Okay, so let's look at why on a genetic level we have to repeat this experiment multiple times and not just one. So we know that a gene consists of two alleles, a dominant allele represented by a capital letter and a recessive allele represented by a lowercase letter. So let's say the seeds that we choose are heterozygous, meaning one dominant allele, one recessive allele, and the dominant allele codes for early flowering. If we crossbreed two heterozygous plants, their offspring will give us something like this. So first, there would be a homozygous recessive gene. So this means it would be late flowering. We will then take the early flowers, hopefully this time homozygous dominant flowers, and crossbreed them. When we do this enough times, we have a greater chance of picking the homozygous dominant genes, and all our offspring will be early flowering plants. And we know that our crops won't have any late flowering plants in them. So early flowering might be controlled by many genes, not just one as an example shown. Thus, we need to repeat this process over many generations to get the results that we need. So we can answer this question by saying that the farmer would first select plants that flower in fewer hours of daylight. They would then cross these individuals to produce another generation. From the next generation, the farmer would again select plants that show the desirable trait and repeat the process for many generations. It's very important to say that you'll repeat it for many generations, otherwise it would not be effective. The next question then states that wheat is grown from seeds. State two conditions needed for the germination of seeds. So for seeds to germinate, we need water as well as oxygen and a suitable temperature. Then on the next page, we have the third question This question says that different types of wheat have also evolved through natural selection. Explain how natural selection differs from artificial selection. So natural selection and artificial selection is very similar to one another. So we can explain this in simple terms. So in natural selection, the selection pressure comes from the environment. It produces individuals that are better adapted for the environment. Natural selection may affect many characteristics and is much slower than artificial selection. Remember, with the wheat example where we used artificial selection, we only tried to change one characteristic, and that is for the plants to flower in less amount of daylight. With natural selection, more characteristics are chosen by the environmental pressures and more characteristics will be altered. Question C then says that scientists can selectively breed for increased pest resistance in wheat. Explain the benefits of growing wheat with increased pest resistance. Now, remember, pest resistance means that organisms like insects, for instance, beetles and borers, those are being chased away or poisoned by the plant so they don't eat on the plant. So this could lead to less pesticides being used. This will lead to less pollution as pesticides cause pollution. When less pesticides are also used, it means that less contaminated food will be produced. This will lead to improved health in humans. And because most pest-resistant plants don't actually kill the pests but just deter them, by using less pesticides, we increase the amount of biodiversity in the area as well. Then we have question 5 on the next page that says phytoplankton are microorganisms that photosynthesize. Question A then asks to state the chemical equation for photosynthesis. So by now, I hope you know this out of your head. So we have 6 water molecules, H2O, plus 6 carbon dioxide molecules, CO2, which would give us 1 glucose molecule, which is C6H12O6, plus 6 oxygen molecules, which is O2. The key word in this question is chemical equation. Make sure to see if they ask for chemical or word equation. Question B then states that a scientist made notes about a marine food web. Now remember, a food web is when multiple organisms interact with one another, so it shows the transfer of energy within an ecosystem. Now figure 5.1 shows the notes she made. So she said that in the sea, phytoplankton photosynthesize. She also says that phytoplankton are eaten by small organisms in the sea called zooplankton. Herringfish and codfish eat phytoplankton. Herringfish also eat zooplankton. Now codfish eat other fish like herring. They then ask us to use the information in figure 5.1 to complete the food web in figure 5.2. Let's start with the first sentence. In the sea, phytoplankton photosynthesize. This means that it's a producer. It uses sunlight to produce glucose. So it would be at the bottom of the food web. And secondly, they say phytoplankton are eaten by small organisms in the sea called zooplankton. So we can see that phytoplankton can be any of the other 3 open spaces. So before we add them, let's go on. Herringfish and codfish eat phytoplankton. Herringfish also eat zooplankton. So we can put zooplankton in this open space as it will eat phytoplankton and then be eaten by herring. Now remember, herring also eats zooplankton. Then they say codfish eat phytoplankton and codfish eat other fish such as herring. So the only block that would have 2 arrows from herring and phytoplankton would be this block being codfish. On the next page, they ask us to state the principal source of energy for this food web. Now the principal source of energy for any food web would be sunlight. The sunlight would be used by producers to produce the first organic material called glucose, and then from there other consumers will follow. The next question then says that Table 5.1 describes features of the food web in Figure 5.2. Complete Table 5.1 by using the information in Figure 5.2. Now, if we look at Table 5.1, we can see they give the feature of the food web, which is the trophic levels, the primary consumers, and the tertiary consumers. They then, in the next column, also tell us the maximum number in the food web in Figure 5.2. Let's look at the first one, trophic levels. So let's go back to the food web and look at the maximum number of trophic levels in the food web. So here we can see that we have 1, 2, 3 trophic levels if we follow this path. 1, 2, 3, 4, 5, 6 trophic levels in this path. 1, 2, 3, 4 for this path, and 1, 2, 3, 4 for this path. So the maximum number of trophic levels would be 6. So we can write here 6. The second one then asks for the maximum number of primary consumers. Now, primary refers to the consumers that eat the producers in the food web. So let's go back again. So here's my phytoplankton. It's eaten by krill, codfish, herring, zooplankton, and then the penguin, meaning that there's 5 primary consumers. Next, we have our tertiary consumers. Now they're in the third consumer group. So here we can see that there's 1, 2, third trophic levels orca, and then codfish is 1, 2, 3 orca again, and then herring is 1, codfish 2, leopard seal 3, orca is then 4. So leopard seal would be 3, zooplankton 1, herring 2, codfish 3, and then penguin, leopard seal, orca 3. So in total we have 3 tertiary consumers. For the fourth question, it says Figure 5.3 is a photograph of an orca. So using the information in Figure 5.2, predict and explain the most likely effect of a decrease in the orca population on the population size of krill and then leopard seals. So if orca would have to disappear and krill is here at the bottom of our food web, then we have to look at what happens in between. So the seal will not be eaten by the orca, so it will increase in size, which means they will eat more krill and krill would decrease. Then for penguins, they will increase as orcas eat less of them, and then krill would decrease as there's more penguins available. So we can answer this question by saying that krill will decrease as the crab-eater seal and the Adelie penguin, which are predators of krill, will increase. Now let's have a look at the impact on leopard seals. So leopard seals are right there below orcas. So as orcas are eating leopard seals, if they are gone, the leopard seal population will increase. So we can say that the population of leopard seals will increase as there will be less predation from orcas. On the next page, question C states that krill and seals can be harvested to produce omega-3 fatty acids. Many people take omega-3 fatty acids as a dietary supplement as they are thought to have health benefits in humans. The first question then asks, list the chemical elements found in all fats. Now, fat is an organic molecule, and all organic molecules contain carbon, hydrogen, and oxygen. The second question then asks, by using the information in Figure 5.2, explain why it is more energy efficient to use krill as a source of omega-3 fatty acids rather than seals. So let's quickly look how the different trophic levels affect the energy efficiency of using them as a food source. So here we have krill. Now krill only eats phytoplankton. Phytoplankton gets all its energy from sunlight. The phytoplankton might use some of this energy, but in the first trophic level, krill would get most of this energy from phytoplankton. The krill will then use this energy for growth. It will also use it for reproduction, movement, and as well as healing, which means that most of this energy is used up and it would need to eat more phytoplankton to be able to reproduce more and to get more energy. This means that the penguin will eat many krill to be able to sustain, and in turn all those many krill would have eaten even more phytoplankton that would have needed to use more sunlight energy to reproduce. This penguin will then use the energy it obtained from the many krill it ate for growth, movement, healing, and reproduction. We then have our orcas that would eat many penguins to sustain themselves. In turn again, the many penguins ate many more krill, and the many more krill ate even more phytoplankton. These orcas then use the energy for growth, for swimming, for healing, as well as reproduction. So if we think about it, if we had to use krill as a source of omega-3 fatty acids, it would be more energy efficient, as not as many krill had to die to sustain the amount of energy needed by the orcas. So we can answer this question by saying energy is lost at each trophic level, as organisms use energy for metabolism, excretion, movement, and reproduction. Now, because krill is at a lower trophic level than seals, less energy would have been lost through the trophic levels. We then have question D that asks us to describe how fats ingested by humans are digested and absorbed. So let's look at digestion as a whole. The first part of all digestion is mechanical or physical digestion. This involves chewing, where food particles are chewed into smaller particles to be easier digested by enzymes, and then also emulsification by bile. This is usually in the situation of fats. Here, bile breaks down fat into smaller tiny droplets of fat, also to digest them easier by enzymes. We have to remember that mechanical digestion does not alter the chemical composition of the biological molecule. Rather, it just breaks it down into smaller pieces. We then have chemical digestion, and this occurs through the use of enzymes. The first enzyme we're going to have a look at is protease. Now, protease breaks down proteins into amino acids. Here we get pepsin as well as trypsin, and the only significant difference between these two is their optimum pH at which they work. One would work in the small intestine and the other in the stomach. Then we also have lipase, which is responsible for breaking lipids into fatty acids and glycerol. Then lastly, we have carbohydrates. Now, carbohydrates breaks down carbohydrates. Now, these are broken down into simple sugars. Here we have amylase, we have maltase, and we have lactase. Now, unlike pepsin and trypsin, amylase, maltase, and lactase do not have different optimum pHs, but they do work on different substrates, meaning that they work on different types of carbohydrates. Okay, let's have a look at the question again. They ask us to describe how fats ingested by humans are digested and absorbed. Now, firstly, fats would undergo mechanical digestion by chewing as well as emulsification by bile in the small intestine. This will increase the surface area of fats for chemical digestion. Fats are then digested by lipase that is secreted by the pancreas. It will break down fat into fatty acids and glycerol. Fats are then absorbed by the lacteals in the microvilli in the small intestine. On the next page, we have question 6 that gives us figure 6.1 that shows a boreal toad, the Anorexis boreas. This toad is listed as an endangered species. They then ask us, A, state the genus of the boreal toad. So if we look at its scientific name, the Onyxurus boreas, we know that the Onyxurus part would be the genus and the boreas part would be the species, which means that the genus of this toad is Onyxurus. Now remember, when writing down a scientific name, we want to underline it. Unlike when we type it, it needs to be in italics, like in the question. Question B then says, one reason for the boreal toad's recent decrease in population is a disease caused by chytrid fungus. State 2 reasons other than the disease that could cause the boreal toad to become endangered. There's many things that can cause an organism to become endangered, including climatic change, habitat destruction, hunting, pollution, new introduced species that are not native to the environment, etc. We then have question C that says that scientists investigated whether increasing the number of beneficial bacteria that live on the toad's skin could provide protection against the fungus. The scientists took 42 toads and placed 21 in group 1 and 21 in group 2. Group 1, the toad's skin was treated with beneficial bacteria, and in group 2, the toad's skin was not treated with beneficial bacteria. The scientists measured the number of toads that became infected with the fungus. On the next page, they give us table 6.1 with all our results. So here in the first column, we have the time since the treatment with the beneficial bacteria in days, and then the number of toads with fungal infection from group 1 and group 2. Now remember, group 1 had the treatment and group 2 did not have the treatment. They then ask us to calculate the percentage of the 21 toads that have the fungal infection after 100 days in group 1 in table 6.1. We need to give our answer to two significant figures, and they give us some space for working. Okay, so we know that we need to work out the percentage, and this is of the 21 toads that have a fungal infection after 100 days. So 100 days, group 1, that would be 13 out of 21. We'll multiply it by 100 to get a percentage, and our answer would be 62%. They then ask us, using the information in table 6.1, describe the effect of treating the toads with beneficial bacteria. So we know that group 1 was treated and group 2 was not treated. If we look at the first 20 days, we can see that the results are exactly the same. Then, for the last 4 groups, from 40 to 100 days, group 1, which was treated, had less toads with fungal infections than group 2. We can also see at the end of the 100 days that overall, less toads in group 1 had a fungal infection. So we can answer this question by saying that treatment had no effect between 0 and 20 days. From 40 days, treatments with beneficial bacteria reduced the number of infections. We can also say that overall, after 100 days, less toads were infected with the fungus if they were treated with the bacteria. Question D then says that there are conservation programs to reintroduce boreal toads into their natural habitat. We need to describe why conservation programs are set up for animals like the boreal toad. So the key word in this question is why. Now remember, the whole point of conservation programs is to keep the organism safe. So conservation programs can maintain or even increase the population size of a species. When the population size increases, it can increase biodiversity in the habitat. This will in turn maintain the existing food webs by maintaining toads as a food source for other organisms. The next question says that conservation programs for other endangered animals such as birds use artificial insemination as part of a captive breeding program. They then ask to describe how artificial insemination is carried out as part of a captive breeding program. So firstly, sperm would need to be collected. This sperm can either be collected from other breeding programs or the same breeding programs. But if we collect it from other breeding programs, it will increase the genetic diversity. The sperm will then be screened and then frozen. The females will then be given drugs to induce ovulation. Now, once ovulation occurs, the sperm is then inserted into the female's vagina.